to Know Before GCSE's GCSE revision for Year 11 Easter revision sessions. Today we're going to be doing some answering of GCSE biology practice questions. So I've compiled a very long document with loads of questions, which we're going to be going through, specifically the hard ones, which a lot of students have struggled with in the past. And eventually, I'm also thinking about releasing some books where I have some personal tips, grade 9 tips, and things like that. So tell me if you would like that. Especially for English, I'll be doing essay plans and quotes and examples of essays and things like that. So do tell me your opinions about that. But anyway, let's get on with the first couple of questions. So as you can see on the board here, the first question is, let's see, explain why only having one ventricle. No, explain how the human circulatory system is adapted to supply oxygen. Okay, so let's talk about this. So let's open the page. So we're asked a couple of things in this question. So how is a human circulatory system adapted to, first of all, supply oxygen to tissues? So let's handle this one thing at a time. So supplying oxygen to tissues, how is this done? So firstly, you need to say the double circulatory system allows high blood pressure and a greater flow of blood to oxygen and to get tissues. So the double circulatory system allows higher blood pressure and greater flow of blood to get more oxygen. Now let's look back a bit at what the question said again. So explain how the human circulatory system is adapted to supply oxygen to tissues. So we've talked about how it allows higher blood pressure and greater flow of blood. We can also say how the pulmonary artery contains and carries deoxygenated blood to the alveoli, where it absorbs oxygen. So the fact that it is a double circulatory system. Oh, so double circulatory system is the first adaptation. The second adaptation comes from the pulmonary artery. The pulmonary artery carries this deoxygenated blood, which I'll represent by minus oxygen, to the alveoli, where it can be reoxygenated. So there are two ways the heart is adapted for sufficient oxygen. Another thing are red blood cells. Red blood cells carry the oxygen. Because of oxygen, what happens is the oxygen combines with the chemical hemoglobin found in the red blood cells to form oxyhemoglobin, and this is what makes the red blood cells red. So the red blood cells carry oxygen, and the fact that it has no nucleus, red blood cells have no nucleus, means that it has a lot of space to carry this oxygen. Now we can also talk about how capillaries carry blood into the tissues, and they have very thin walls to allow for optimum diffusion of oxygen. So we can talk about capillaries. Now capillaries are mostly discussed when we're talking about the small intestine and the villi and how it is able to absorb all the nutrients from there. But capillaries also are used for putting a lot of substances into the blood. So they have thin walls, elastic, and this allows for optimum diffusion of oxygen. Okay, let's move on to the next question now. So the next question Explain why having only one ventricle makes the circulatory system less effective and less efficient than having two ventricles. So what happens is, firstly, we looked at already the purpose of ventricles. So the purpose of ventricles is to carry that deoxygenated blood. So firstly, you need to identify what the purpose of the ventricles is. The purpose of the ventricles is to carry this deoxygenated blood to the lungs to go and get oxygen. Now imagine what would happen if we didn't have ventricles. That's basically what this question is asking us. So if we didn't have ventricles, this means that the oxygenated blood and the deoxygenated blood would then mix. This means that less oxygen will reach the body cells. Because remember, if they're going in two different directions, because the oxygenated will be going to the body cells and the deoxygenated will be going to the lungs. Now, because they're going in two different directions, as the oxygenated would try and get past the deoxygenated, it would cause friction, waste energy, and it would take a lot longer for the oxygenated to get to the blood cells, the body cells, and the deoxygenated to get to the lungs. Therefore, mixing it would be a bad idea, and that's why our body has adapted against it. Let's move on to the next question. The next question is, explain how human lungs have adapted for efficient exchange of gas via diffusion. Okay, so let's look through this. So how has human lungs adapted for efficient exchange in gas via diffusion? So firstly, this question will force you to think about everything that comes in via diffusion. And we know that in terms of alveoli, the alveoli brings in oxygen via diffusion. Capillaries as well. Now the alveoli bring in the oxygen via diffusion, and then it goes to the capillaries again via diffusion. And then we have the breathing mechanisms and the diaphragm, which move air in and out. That is technically diffusion. And then there's also the fact that there's a large capillary network. The fact that there's a large capillary network means that this increases the surface area to volume ratio, and therefore more oxygen can reach. Okay, let's move on to the next topic, which is explain why a stent prevents a heart attack. Okay. Oh, hello there. Just seeing your comment. If you have any questions, feel free to ask, okay? So we're going over the question, explain how and explain why a stent prevents a heart attack. So, do. Stent, the function of the stent is to keep the coronary arteries open. Now, by keeping them open, this then allows blood to flow through it. And as blood can flow through it, this means that glucose and oxygen can get to the heart. So glucose as well as oxygen can reach the heart. And because they can reach the heart, this means that the body can undergo aerobic respiration, and therefore you can breathe and make energy. And that is how a stent prevents a heart attack, because a heart attack is when you can't get blood to flow through because the glucose and oxygen can't reach the heart, and therefore aerobic respiration cannot take place. Okay, moving on to the next question. So the next question is, list the risk factors of coronary heart disease. So people often get confused. What do risk factors mean? Well, let's look at that now. So risk factors are anything that could cause whatever the disease that is discussing about. So in terms of the risk factors of coronary heart disease, let's look at that. Risk factors include things like lack of exercise, obesity, smoking, stress is even one, and things like that. So these are risk factors of coronary heart disease because they could cause coronary heart disease. Diabetes, smoking, too much fatty foods. Perfect, that's perfect. Great answers there. Okay. Let's do the next question. So the next question is, evaluate the use of cholesterol blockers versus statins for a person with high cholesterol. So normally with questions like this, they would give you a bit of background information regarding what are cholesterol blockers, what are statins, and what are the functions. So let's just look over that now. We have cholesterol blockers, cholesterol blockers, and then we have statins. So statins are drugs that you have to take life. The moment you start taking them, and in the UK you're given them immediately at the age of 50. So that's what statins are, drugs you have to take for the rest of your life. Now, cholesterol blockers you don't have to take. So you don't have to take for the rest of your life. Now, statins can also damage organs because of the heavy amounts of drugs. So statins can be a bit dangerous because they can damage organs. However, cholesterol blockers do not damage organs. Statins can also cause death, but cholesterol blockers don't. Statins can reduce cholesterol to zero. So statins can totally eliminate cholesterol, whereas cholesterol blockers can only reduce it. So cholesterol blockers only reduce, whereas statins can eliminate the prospect of any cholesterol. Okay, and if you mention a couple of these points, then you will definitely add up to 6 marks. Okay, moving on to the next question. So, what condition may be treated using an artificial pacemaker? So, just in case you didn't know, a pacemaker is the thing that controls the heartbeat, the regularity of the heartbeat. So, in terms of conditions, we could just say an irregular heartbeat, which has a proper name for it, but just say irregular heartbeat for this one. In terms of if you want to know the actual name, I cannot pronounce it, but I'll write it down for you here. This was in the mark scheme, so that is a full name for it, if you wanted to write that down. Okay. Now the next question. Compare mechanical valves to biological valves. So again, this would be a similar type of question where you will be given some sort of information. So Mechanical valves versus biological valves. Okay. So firstly, you need to make sure that you know what a valve is. What a valve is. So a valve is the thing that's found in the heart that prevents the backflow of blood. The aim is for the double circulatory system is that they flow in different directions. And in order to keep each one in the same direction, we have valves. That is correct. Biological valves can last longer. That's correct. So let's write that. Biological valves can last longer. Feel free, anyone else, to chat what else they think we can compare. So biological can last longer. Another thing is that biological doesn't require additional medication. So no additional medication. The reason why is because your body can more easily get used to biological. For example, like a pig valve rather than a mechanical, which is machinery. Mechanical usually has to get replaced. That is true as well. So mechanicals require replacement. Although biological does require placement as well, but mechanical has a shorter lifespan. Now, because of mechanical and the metals and materials that are in the mechanical, you also need medicine called anti-clotting because it can cause clots that can be really damaging. So you need the medication anti-clotting, anti-blood clotting medication. And also another thing is that mechanical, the valve may be harder, but with biological, it will be more softer tissue. But that's not really too much to say. And the final thing is with mechanical, it can also lead to excess bleeding after you've put it in. So it can be quite dangerous. Whereas biological doesn't tend to excess bleeding. Okay, let's move on to the next question. Well done for your thank you for contributing, everyone who did. Okay, so the next question is, where are we? Explain why a person with a leaking heart valve has a difficulty exercising. So let's just read that again. Explain why a person with a leaking heart valve has difficulty exercising. Okay, so we already know the purpose of valves. They prevent the backflow of blood. So if it's leaking, this means that the blood is not doing its function. Some of the blood is backflowing. And why would they then have difficulty exercising? So these are one of the questions where you have multiple steps in order to answer this. So let's begin. So obviously, someone with leaky valves, that's what they're called. Leaky valves cause the backflow of blood. Now, what does this then mean if there's a backflow of blood? This means that less blood gets pumped around the body. Okay, so if less blood gets pumped around the body, then all of the substances that blood carries will not be able to be supplied to. The blood from the heart will not supply enough oxygen to working muscles. That is perfectly correct. So, because less blood gets pumped, less oxygen gets supplied to the muscles. So less oxygen to the muscles, and therefore less aerobic respiration. And because there's less aerobic respiration, this means that there's less energy produced. And if there's less energy produced, this means that there's less efficient muscle contraction. So basically, a summary of what you wrote. Less efficient muscle contraction. And then you can also talk about how anaerobic respiration will start to happen, and this will cause an oxygen debt and then the buildup of lactic acid. But that is definitely going a bit above and beyond. This is all you have to say. If you say these steps, then you'll definitely get enough marks. Okay, so I think turn the page. Yeah. Explain the risks of a pregnant woman smoking and drinking. That's the next question. So, what are the risks of a pregnant woman smoking and drinking? Okay. So here we have to think about what smoking has in it and what alcohol has in it that can be dangerous. So let's talk about smoking, and then we'll talk about alcohol. So firstly, smoking contains carbon monoxide. Now, carbon monoxide is really toxic because of the fact that carbon monoxide is a toxic gas which reduces the amount of oxygen in the blood cells. And if there's less oxygen in the blood cells, this means there's less oxygen for the fetus. So it can increase the risk of miscarriage because of the fact that the infant can't breathe. Now, someone said carcinogens. Yes, that's perfect. So carcinogens are things that cause cancer and increase risk of cancer. So smoking, and this not only risks the fetus's life, but also not only the pregnant woman getting cancer, but also the fetus. The fetus can be badly affected from the cancer. Okay, alcohol. Let's talk about alcohol. So, alcohol within the first 3 months can actually increase the risk of having a premature child or having a miscarriage. So, having alcohol within the first 3 months increases the risk of being premature or miscarriage. And alcohol within beyond this thing, so beyond 3 months, then means that it can lead to fetal alcohol syndrome, FAS. Okay, so they're the things you need to say for this question. Now let's move on to the next question, which is describe effects of liver failure on the human body. Okay, so let's look at liver failure. So, first of all, this is all to do with a lot of different topics here, combining it. A bit of homeostasis and a bit of this. So, firstly, we can say no bile is produced. So, what is bile, first of all? Bile is the chemical that emulsifies lipids. Emulsifies, if you didn't know, just means converts the big lipid droplets into smaller droplets so the body can more easily absorb it. So when there's no bile produced, this then means that there's less fats digested, and also if there's no bile produced, pH conditions from the acid will remain acidic. So because the food comes from the stomach to the small intestine. Now, bile intervenes at this point here, and bile allows the pH, restores the pH from the acidic conditions in the stomach to become neutral in the small intestine. Now, if we don't have bile, this means that the pH conditions would remain acidic. Now, this is problematic because if the pH conditions remain acidic, this then means that the enzymes will not work effectively. The reason why they won't work effectively is because the pH are their optimum conditions. Okay. Now, another thing, so that's all about bile. Another reason is the fact that there's because lactic acid is produced when you have anaerobic respiration and it goes to the liver. Now, if there is liver failure, then the lactic acid is not broken down. And what happens if the lactic acid is not broken down is that the accumulation of lots of lactic acid is actually toxic and the body can be poisoned. Due to the accumulation of lactic acid. Now, another thing that happens in the liver is that the proteins are deaminated, or the amino acids are deaminated, sorry. And because the amino acids won't be deaminated, then that means that ammonia will form, and ammonia is very toxic. Okay, so there are 3 different reasons why. I agree with the jaundice comment. However, what they would want is they would want you suggest how that could be caused by biological, in terms of biologically speaking. But if you can, that's perfectly a good suggestion because that is one of the causes. Okay, let's look at the next question. Explain how diabetes can cause the cells to lose more water. Okay. So let's just think about that. Explain how diabetes can cause cells to lose water. Okay. This question, you have to think about concentrations and things like that. So, when you have diabetes, what happens? When you have diabetes, this suggests that you don't have much insulin, or your body isn't responsive to insulin. If your body isn't really responsive to insulin, this means that you have a lot of glucose in your blood. If you have diabetes, you have a lot of glucose in the blood. Now, because there's loads of glucose in the blood, it means that the blood is very concentrated. And there isn't enough space because the glucose is taking it. So what happens is the water moves out of the blood, out of the blood cells, via osmosis. So this is sort of a hard one to get your head wrapped around. But what it's just saying is that because you have diabetes, you have more glucose in your blood. So your blood is very concentrated. So the water moves out of the blood cells via osmosis. So you can carry all of this extra glucose. Okay. So just ignore those two questions just beneath that question because that doesn't really make sense, and we need to move on to the next topic now, which is cancer. So the first question, compare between benign and malignant tumors. Okay. So I'll give you a bit of time to just take that question in, and if you have any answers, then feel free to put it in the comments or the chat. Okay. So, the similarities and differences between benign and malignant tumors. So, let's look at the similarities first, and then we'll look at the differences. So, in terms of similarities, for both of them, they are both uncontrollable growths. And they are both formed from a person's own cells. Yeah, that's correct, by the way. Benign tumors do not spread and are not cancerous. That's correct. So, in terms of a difference, benign tumors are enclosed within a membrane. Enclosed within a membrane. And because it's enclosed within a membrane, it does not form secondary tumors, so it does not spread and therefore is not cancerous. Whereas malignant is not enclosed and therefore it does spread, forming secondary tumors, and that is what we call cancerous. So that is very correct. Okay. Benign is slower than malignant. Benign is slower than malignant. I guess you could say benign is slower than malignant, or more that it's just it's limited to being in its own tissue, that it can't really spread. Malignant tumors can spread to other parts of the body as they're cancerous. Yeah, that's correct. Okay. And yeah, that's all we can say about this question, really. But that's why they will never ask you more than a 4 marker on this, because there's not more than 2 points worth. Okay. Now let's move on to the next question. So the next question is, name some risk factors of cancer. So you can put them in the chat. What risk factors are there of cancer? What things cause cancer? Okay. So risk factors of cancer. Firstly, we have things like carcinogens in smoking, as you guys mentioned in the chat. So carcinogens. We also have alcohol intake. And if you have alcohol, we're going to have smoking as well, which I guess is linked to the carcinogens comment. UV radiation, that is a fair comment. And you can also say diet, because diet is linked to cancer. Yeah, radiation. In terms of genetics, some fully allow that answer in because they don't say that it's a risk factor, it's more of a inherited trait. But so just try not to say genetic unless you have nothing else to say, because they sometimes won't allow it. Okay, moving on to the next question. Smoking can cause diseases such as cancer. Explain why this is not a communicable disease. Okay. So what this would require is just 2 statements talking about how smoking would not cause a communicable disease, and how cancer is not a communicable disease. So, cancers cannot spread to other people. It can to or from different people, because remember, it's formed by a person's own cells. And another reason why we'll say this is that cancers are not formed from a pathogen. That's another way of saying it. So one is cancers cannot spread to different people, and another thing is cancers are not caused by pathogens, but by our own body cells. And you can get 1 mark for each one for that question. Okay, moving on to the next question. So, Helicobacter pylori are acid-tolerant bacteria which can damage the human mucous lining. Suggest how an infection might result in a stomach ulcer developing. Now, this is a very tricky question because they're asking us about a lot of concepts which are unfamiliar to us and to you guys being GCSE students. However, do not fear, because you can easily break this question down. Okay? So we're saying that we've got a bacteria here, and this can damage the human mucus lining. And we're told that it's acid tolerant. So how can this result in a stomach ulcer infection? So what do we know about the stomach, first of all? Stomachs contain hydrochloric acid. Now we're told that this special bacteria is resistant to this acid. Therefore, if we know that it's resistant to the acid, we then know that hydrochloric acid could then cause an ulcer. The hydrochloric acid, or more the bacteria, can then cause an ulcer in the stomach because it's not protected from the hydrochloric acid because it's resistant to it. Describe how a person infected with Helicobacter pylori could also develop liver cancer. Okay, so now we have to connect this idea to livers. So what do we know about liver? Well, we know that this bacteria, we know that cancer in itself, if malignant, will spread to other organs. So they can move via the blood, forming a secondary tissue. So what it's just saying here is don't focus too much on the bacteria part of it. Focus on the fact that if it could develop liver cancer, then that means that the cancer would be malignant. So it would have to spread from the stomach to the liver via the blood. Okay. Then on to the next question. Let's move on to the next page. Define and explain the process of transpiration. So let's look at that now. What is the process of transpiration? Okay, let's look at the steps. So firstly, we have we have to define and explain. So let's define it first. So in terms of defining it, transpiration is the movement of water from the roots to the shoots via the xylem tissue. it's key that you mention this part via the xylem tissue. Okay? So transpiration is the movement of water from roots to the shoots via the xylem tissue. Now, if we talk about the steps of transpiration, so let's start with talking about the evaporation. So the evaporation occurs in the leaves. So what is evaporating here? Water is evaporating in the leaf from inside the cells. It diffuses through the air spaces and out through the stomata. So the water diffuses through the air spaces in the leaves and then out through the stomata. So that's how the water exits. Then we have to talk about how it then enters. So water then passes from the xylem tissue to the leaf to replace this lost water in the leaf. So water then moves from the xylem tissue to the leaf to replace this lost water. Then what happens is water is then drawn up from the root hair cells to the xylem cells. To replace the lost water in the xylem. And it always initiated by the fact that the evaporation occurs in the leaves of water. The water going through the air spaces and out through the stomata causes all of this. Okay, so now let's move on to a question about bacteria culture. So we're now moving on to B3. So explain how... Wait, is this the right page? Okay, so explain how to prepare a bacteria culture. Now this will cross over in the practical side of questions. So let's go over that now. Explain how to prepare a bacterial culture. So let's look at the first thing. We have the different stages of preparing a bacterial culture. Let's call them, the first one, pre-inoculation. Then we have inoculation, which is when we have the inoculating loop that's putting the whatever substance onto the petri dish. And then you have post-inoculation, what you do afterwards. So the three different stages. So what happens at pre-inoculation? Let's look at that first. So at pre-inoculation, what happens is, first of all, we have to sterilize everything. Things we sterilize include things like the petri dish itself, because we want to get rid of any unwanted bacteria, because we don't want them to compete with the bacteria we are trying to grow and therefore cause problems. Now we also want to sterilize the inoculating loop. How do we sterilize the inoculating loop? What we do is we pass the inoculating loop through a fire and as it goes through the fire, it then kills any unwanted bacteria. So sterilize the petri dish, sterilize the inoculating loop through a flame. Now we get to inoculation. So what happens during inoculation? During inoculation, you then use the loop to spread the bacteria across the surface of the petri dish. Now, there's certain methods you do now for post-inoculation, because that's the inoculation part. So for post-inoculation, there's certain things you need to keep. So firstly, you want to tape the lid onto it, but then leave stops air can enter. The reason why we want air to enter is so that it undergoes aerobic respiration, because if it was to undergo anaerobic respiration without air, this means that it could lead to really harmful bacteria starting to grow. And we don't want that from occurring. Okay. Another thing that we do is we turn it upside down. So when we're storing the Petri dish, we store it upside down. Now, the reason why we store it upside down is so that if there's any humidity or wetness, it doesn't drop down onto the dish. So any condensation. So instead of the condensation dropping down, if you turn it over, then it can't drop down, it will stay on the surface. So it prevents condensation from messing up the process. And another thing, you have to have certain temperature range in order to store this bacteria. So you must store it between a maximum temperature of 25 degrees Celsius. You don't want anything higher than that, because higher than that, really harmful bacteria can start to grow. And if it was lower than, say, probably 10 degrees Celsius, then not much would grow, because the bacteria would just freeze. So that would be counterintuitive. Okay, so that's the bacteria culture. Now let's talk about some viral diseases. So explain why TMV causes stunted growth. TMV is tobacco mosaic virus, by the way, which is for plants. So explain why tobacco mosaic virus causes stunted growth. Now, how are we going to answer this? TMV causes stunted growth. So what is needed? What nutrients is needed for plants to grow? Well, we have nitrates, which are formed for the production of proteins, and then we have magnesium, which is used in chlorophyll. So what happens is, because of TMV, these black spots appear on the leaves. Now, because these black spots appear, this reduces the amount of chlorophyll that can then absorb the sunlight. Lack of green pigment, chlorophyll, sunlight, mineral ions. Perfect. Yeah, that's perfect answers. So black spots, lack of chlorophyll, therefore, if there's lack of chlorophyll, it lowers the rate of photosynthesis. And if it lowers the rate of photosynthesis, then less energy is being made, and then less energy can be converted for growth. So that's how that will be. The next question is also about viruses. Explain how measles spreads from one person to another. So they would have expected you to do some research within your GCSE biology and know a bit about all of these diseases. So let's talk about measles now. So measles is a viral infection, and it's spread by droplets in the air. So in order to spread it, you need to have inhaled droplets in the air that contain measles. And where do these droplets come from? They could come from sneezing, it can come from coughing, touching certain surfaces, and things like that. Okay, let's now look at bacterial diseases. Okay, so TB is a bacterial infection. So this is tuberculosis. Explain why people living in a densely populated area are more likely to be affected by TB. So what they're asking here is what is the correlation between the population in which you live in, how densely populated, how many people you live in, and how likely something is to spread? So how do we connect those two ideas? Firstly, we can say because there's a greater population, so due to the greater population, this then means that there's a greater chance for there to be TB droplets in the air. And because there's a greater chance for there to be TB droplets in the air, there's a greater chance for more people to be infected. So people to be infected by tuberculosis. It's an airborne virus. If there are more people in an area, you're more likely to get infected. Perfect. That's perfect. Okay, let's move on to the next question. So the next question, where are we? The next question, give reasons why a person infected by a small number of tuberculosis may not develop the disease. So why, if someone was infected by a small amount, how would they not have the disease? So what we can say here is that the white blood cells, sorry, I'm not showing my screen, am I? So the white blood cells produce enough antibodies Remember, antibodies work against the antigens. So against the antigens of the TB. So the white blood cells produce enough antibodies, which then rapidly destroy the pathogen. So in this case, rapidly destroy the TB. And that means that, so this happens before you feel any symptoms. Now, the reason why we actually feel symptoms, let me just explain this because it's not actually explained in many things. So the reason why we feel symptoms is because of toxins. Toxins are things that pathogens produce. And what happens is these toxins damage and actually destroy our cells. So when we feel ill, this is actually the toxins' work of damaging and destroying our cells, and that's why we get symptoms. So if you were ever asked, why do you get symptoms from diseases? You can say, the pathogens release toxins which damage and destroy our cells, and that is how we feel symptoms. Let's move on to the next question. So, describe how plants become infected with rose disease. Okay. So this is talking about the transmission of plant fungal diseases. Okay. So firstly, let's just mention here, rose black spot is a fungal disease. Don't be afraid to state the obvious as well, because if they don't mention it in the question, then they probably expect you to mention the answer. So rose black spot is a fungal disease, and what you can say is its spores spread, and you should know this from your revision over rose black spot, is it spreads through wind and in rain droplets. Okay. So it's a fungal disease and its spores spread through wind and rain droplets. So the reason why we mention that it's a fungal disease is because fungal diseases spread via things called spores. Okay, now let's look at the next question. So, what could a gardener do to reduce the risk of rose plants getting infected with rose black spot disease. So feel free to comment, what could the gardener do that will reduce the risk of plants being affected by rose black spot? Okay, I'll write some ideas up on the board now. So there are some connection issues, so do tell me if you can still hear me or if there's a bit of delay, because I'm being told there's connection issues. Insecticides. Yep, that's really good. So insecticides. So firstly, we could say removing all of the affected leaves or plants. Okay, so removing all infected leaves and plants. Okay, thank you for the confirmation. Insecticides, as you said. We could also say treat the plants with fungicide. The fungicide will work against the fungus, and you can also say grow plant resistant varieties. So grow plants that are resistant to rose black spot. Okay. So the next question, which is evaluate the reduce the spread of malaria. Now this is a 6 marker. So what you information about the different. Okay, I'm so sorry for that. I hope you can still hear me. And there's been a lot of issues going on with this. Everything has just gone, so let me go back and get them back for you. Oh, we were approaching the end of the session anyway, so if you want me to do more of these sessions, please tell me, because I am all up for requests and I'll definitely listen to you if you have anything to tell me. But yeah, that was a bit of biology paper 1. In case you wanted to know what questions we didn't do, so you could do them in your free time, we didn't do things about protists, we didn't do vaccination and monoclonal antibodies, and we didn't do things about the discovery and development of drugs. And then obviously all we had left to do was B4, which is photosynthesis, respiration, energy, and things like that. And then, yeah. Uh, yes, those answers are correct as well. Vaccine, malarial nets, insecticides, education, programs which warn people. Also, they say in terms of vaccine as well, some extra information. Also, have the vaccine 2 weeks before you plan to fly out, so it starts and it's already in your body, and your body is already familiarized with it. But yeah, so do ask me if you have any other requests. But yeah, we've practically finished biology paper one, so you've done great because it's already 8 o'clock and you've already done some revision. Oh, thank you, everyone. That makes me feel so happy. I'll definitely do more then. So, would you like me to do more for each of the sciences? I wouldn't mind doing it for other subjects as well, like geography, English, things like that. So, feel free to just tell me, and then I'll definitely do them if you want them. But thank you so much. Okay. Thank you for your request. I'll definitely write that down now, but I'll leave you alone. Well done for your revision, and keep out because I'll start scheduling the what's it called, live streams for the different questions, and then you can just make sure that you attend them. And there'll be like similar times to this one today, and we'll do the rest of the sciences. Okay. Thank you for watching, and see